Hey guys, this is Bernadette Hartney,
And Mary Kozik.
and today we are going to be telling you about salicylic acid.
Okay, to start off, I'm going to give you guys a little background information about salicylic acid. We did research on NCBI from an article called Dietary Salicylates and from PubChem in an article called Salicylic Acid. To start off, salicylic acid is part of a diverse group of phenolic compounds and has an aromatic ring with a hydroxyl functional group. It is a natural compound which is made in plants, specifically the white willow, which you can see on the picture on the slide, and white wintergreen leaves are known for their production of salicylic acid. The active extract of the willow bark is called salicylin, which you can see a picture of on the slide next to the willow tree. That is the structure of it. When it was first isolated by a chemist named Johann Andreas Buchner in 1828 and later researched by Henry Lewarx in 1829, who further isolated the compound. Salicylin has been later found to be an anti-inflammatory reagent in the body and was the historical origin of what we know as aspirin. Raphael Perea, an Italian chemist, was able to convert salicylin to a sugar and a second component, which on oxidation becomes salicylic acid. Therefore, salicylic acid was derived from the metabolism of salicylin. Overall, salicylic acid is a phytohormone, which means this is a plant hormone, which affects plant growth and development, photosynthesis, transpiration, and ion uptake and transport. Salicylic acid is involved in endogenous signaling, and it serves as a function in plants and a defense against pathogens. Humans can metabolize salicylic acid in large quantities by eating plants. One study has found that vegetarians who are not taking aspirin had urinary levels of salicylic acid higher than those of non vegetarians. This led to the conclusion that vegetarians are getting their salicylic acid from plants they were ingesting.
So now that you know a little bit about
Okay, so now that you guys know where you can find salicylic acid, let's talk about the biosynthesis of salicylic acid. So, after doing a research on keg.com, we found that the entire biosynthesis pathway has many different starting points, but we decided to show you guys the starting point from phenylalanine because it is a compound that we are all very familiar with. Okay, so as you can see from the figure, on the left you see dashed lines, and right over to the right of those 4 lines, is phenylalanine. So we have to follow phenylalanine, and it gets converted into trans-cinnamate, which you can just follow the arrows to the right and then down, and then trans-cinnamate is then converted to benzoate, and then we go up from benzoate to salicylic acid. This reaction has been shown to occur in 3 distinct reactions. Again, phenylalanine to trans-cinnamate to benzoate to salicylate. As you can see on the dotted figure, there's a dotted line going from trans-cinnamate to benzoate. This dotted line indicates that they do not know exactly how this reaction occurs because they do not know the exact reaction that catalyzes it, but we have proposed a mechanism for you that Bernadette will be going through. Reaction 1, again, in case you guys didn't remember, the reaction first starts off from phenylalanine to trans-cinnamate and is completed by phenylalanine ammonia lyase, which we refer to as PAL. Reaction 2 goes from trans-cinnamate to benzoate, and like I said before, this reaction is not specifically known how it is carried out, but there's a proposed mechanism, again, that Bernadette will go through. And then reaction 3 is from benzoate to salicylic acid, and it is completed by benzoic acid to hydroxylase, and which is referred to as Ba2H, and we will not be going through the synthesis of that one.
So these are pictures of the compounds that are converted from and to respectively. Like Mary said, you start with phenylalanine on the left. PAL then converts it to tranexamic acid. Then it goes from tranexamic acid to benzoic acid by an unknown enzyme. And then it goes from benzoic acid to salicylic acid by Ba2H. And next we're going to show you the mechanisms of how the first two get converted. So this shows you what we are losing in the reaction from phenylalanine to tranexaminate. This shows you the most important step in the enzyme PAL, which has to do with an MIO cofactor. MIO stands for 4-methylidine imidazole 5,1 cofactor. PAL belongs to a family of enzymes that need that cofactor for the conversion of phenylalanine to tranexamic acid. And under conditions of high ammonia concentration, the deamination reaction is actually reversible. So these enzymes are used very frequently in research. So as you can see in this picture, there's a deamination reaction which occurs when the amine group attacks this MIO cofactor and the electrons are pushed all around MIO. And then when another enzyme comes in and deprotonates, that kicks off the amine group, which then is attached to the MIO cofactor. And you'll see the full step mechanism in the next slide. So this slide shows you all of the amino acids in the active site of PAL. And so you have 2 tyrosines, an arginine, which is pretty much just used for stability, and then your MIO cofactor. So how this reaction proceeds is the tyrosine first deprotonates the ammonia, and once it's deprotonated, it attacks the MIO cofactor. So you see this in the intermediate 1 stage. Moving on to intermediate 2 stage, then that amine attacks the MIO. Oh, sorry, that happened in intermediate 1. And in intermediate 2, you see it connected, where then the other tyrosine will deprotonate that hydrogen. Its electrons will collapse down. That arrow is not shown, but you can see what would happen, which then would kick off the amine to be attached to the MIO cofactor. So going down on the bottom left to intermediate 3, we now have formed an alkene, and we have our amine group on the MIO, which then gets the hydrogen off of the above tyrosine, and once it is NH3, it wants one more hydrogen from the other tyrosine, at which point it becomes ammonium, and so you're left with your products where it has the enzyme reset back to normal, ammonium free floating, and we have our product of cinnamonic acid. Next, we need to convert from cinnamonic acid to benzoic acid, and sorry, this actually goes right to left. This again was taken from keg.com, which told us the reactants and products and what comes in and what comes out, but we had to generate a reaction mechanism for this, which I'll explain in the next slide. So here is our proposed mechanism. We have our tranexamic acid in the active site of some unknown enzyme, but we propose some acid-base chemistry with an NAD in here. You have a base come in and deprotonate water, then that hydroxide attacks, which those electrons, attacks the alkene. The electrons from the alkene get protonated. The NAD comes in, and next you have the base deprotonate the hydroxyl group, which just attacked. The electrons collapse down to create a carbonyl, and that hydride attacks the NAD, kicking the electrons around the ring on NAD, and then your NADH leaves. Water again comes in. Now we're in step 3 on the right side. A base deprotonates the water. Hydroxide attacks the carbonyl, kicking the electrons onto the oxygen, which you can follow down. Those electrons collapse back down and kick off acetic acid in the process, and we are left with our benzoic acid and acetic acid. Lastly, our benzoic acid needs to be converted to salicylic acid, and this is done by the enzyme BA2H. Like we said, we don't have to explain this mechanism because you already got to learn about 2 great ones.
Okay, so now that you saw all of the fun mechanisms of salicylic acid, now let's talk about how it is regulated. So I did research from NCBI about the biosynthesis of salicylic acid in plants, and from Cell Press Journal article. So, once synthesized, free salicylic acid levels can be regulated by a variety of chemical modifications. Many of these modifications indicate salicylic acid. However, some have properties that may aid in long distance salicylic acid transport or the activation of stress responses complementary to those induced by free salicylic acid. In addition, a number of factors that directly or indirectly regulate expression of salicylic acid biosynthetic genes or that influence the rate of salicylic acid catabolism have been identified. However, the main regulator that I'm going to be talking about, and the most research that I have found on it, is the inhibition of the enzyme PAL, which we showed you the activity of above. And this inhibition is created by trans-cinnamate and benzoate. So the first one I'm going to talk about is how trans-cinnamate inhibits PAL. So phenylalanine aminolyase is the first committed enzyme in the pathway that synthesizes phenylalanine to trans-cinnamate. There are many regulatory mechanisms at multiple levels that control the transcription and enzymatic activity of PAL. So, first of all, PAL activity, gene transcription, is negatively rated by trans-cinnamate. In this way, trans-cinnamate acts as an inhibitor of PAL, and it also blocks the downstream pathway of cinnamate 4 hydroxylase, otherwise called C4H, which you can see on the diagram to the right. So this scary diagram with all the circles and everything, you see PAL, and the C4H is connected to it, and so when these are connected, they are both making trans-cinnamate. So PAL is inhibited when there is an overexpression of trans-cinnamate by C4H as well. So going down the pathway, excessive trans-cinnamate can directly inhibit enzyme activity itself, or it can even impair PAL transcription. So PAL transcription, if there is a lot of trans-cinnamate in the pathway, the transcription of PAL will even be deregulated. So when this happens, PAL is downregulated, then phenylalanine cannot become trans-cinnamate, which is the second step in the biosynthetic pathway. Another inhibitor of PAL that I found on Brenda was the accumulation of benzoate in the biosynthetic pathway. And so to show why this would be bad, so phenylalanine goes to trans-cinnamate, then to benzoate. So if we have an accumulation of benzoate, this will decrease the activity of PAL, and then PAL will be inhibited. Then you cannot get to trans-cinnamate, so the whole biosynthesis of salicylic acid will be decreased.
So this concludes our presentation, and we hope that you have enjoyed enjoyed learning about salicylic acid. Make sure you eat some veggies so you can reduce inflammation.
Yay.